Hello, this is Berejnoi, and in this presentation I'm going to be talking about gene drives and ethics associated with it. Let me start with a story that began 150 years ago with Gregor Mendel. He was an Austrian monk and biologist who established that offspring have a 50% chance of inheriting a gene from one of their parents. This laid the basis of modern genetics and understanding of how genetic inheritance works, including the concept of selfish genes, meaning that a gene promotes its survival by being passed on to offspring. And this concept is still being used today. Later on, in the 1960s, this man, George Craig, and mosquito biologist and his colleagues suggested to use mosquito breeding programs to manipulate their genes to produce only male offspring. This was a mechanism to control population of mosquitoes. The problem was that the technology didn't exist back then. They had to wait another 30 years for this to be possible. And later, still with this idea in mind of how selfish genes work and inheritance, this man, Austin Bart, an evolutionary geneticist, had a question. He wanted to know if an insect's genes could be not manipulated so the insect wouldn't spread disease anymore. He proposed using a selfish gene that drives genetic changes into the natural population in mosquitoes. With the existing technology, genes could be manipulated, but they couldn't be passed on to offspring. However, that changed with the introduction of a new technology called CRISPR-Cas9, which allows to make a change in the DNA of a species, of a mosquito in this case, which will be passed on to offspring, and also makes a gene immune to viruses. Here we see it. With gene drive technology, the inheritance of a gene is favorably biased. This means while in normal circumstances a gene has a 50% chance of being passed on, which is the principle that Gregor Mendel established, with a gene drive, in theory, it has a 100% chance of being passed on. And we see it on the right, gene drive inheritance, how the mosquito engineered with gene drives is being passed on to offspring. Gene drive technology works well with sexually reproductive species that reproduce rapidly. An example is insects and rodents. This technology has new advances every day with a significant contribution of the Bill and Melinda Gates Foundation of $75 million, which is expected for use by 2029. So we see that the idea of gene drives started with mosquitoes, and currently is still working on mosquitoes. Scientists are trying to find ways to reduce diseases that mosquitoes cause, such as malaria, dengue, Zika, chikungunya now in South America. And the research has also expanded to other areas, to other species. One case is to control the population of mus musculus, which is an invasive species of rodents. It's a mouse that populates in many islands in the world, and actually 80% of islands have invasive species that are affecting native species. So with this, the idea is to skew the ratio of sexes, to have more males and females by engineering a gene with gene drives, so the mosquito only has male offspring. And at some point, there won't be any more female offspring, making male offspring go extinct at some point because they won't have any more females to reproduce with. The other example is with Palmer amaranth. It's a crop and a weed, or both, depending where in the world you are. In the American South, it's a weed. So what scientists are trying to do with these sexually reproductive species is to have more male flowers than female flowers. So at some point there won't be any more female flowers, and male flowers are not going to be able to reproduce, making the species go extinct. Let me talk more about mosquitoes. The problem with mosquitoes is that they affect populations in tropical and subtropical regions in the world. Their diseases affect people in these areas. Developing countries in this part of the world, and also the most vulnerable populations are affected. In Africa alone, 300,000 children die of malaria each year. With dengue, Zika, chikungunya, there are also many deaths annually that affect the people, and there are measures being applied to every part of the world to see if they can reduce deaths. For example, the World Health Organization endorsed the use of the first ever dengue vaccine in 2016, created by Sanofi Pasteur. The problem with methods like this is that not everybody has access to them. Sometimes people are not aware of resources available to them. Sometimes they live too far away from hospitals to have access to these resources. So with GeneDrive, the goal is to make female mosquitoes, who are the ones who bite, not able to spread disease. That is because the virus wouldn't be able to live on the mosquito. It would die immediately. That's one solution. The other solution is skew the ratio of sexes. The same case as with mice and with Palmer amaranth. Having more males than females is going to make, at some point, the species would go extinct, because the males wouldn't have any more females to reproduce with. With this, people wouldn't have to worry about diseases anymore. It would be a huge benefit to humanity, because there wouldn't be more diseases caused by mosquitoes. Now the other side of the ball. We live in an imperfect world with imperfect humans that create imperfect technologies. Remember that at the beginning I mentioned that with GeneDrive, in theory, the gene is being passed on to offspring 100% of the time. We don't know, however, if that is going to be the case in the outside the lab, because right now in the lab it works in theory. What happens when the virus waits a few generations of offspring for the mosquito? Maybe it can evolve, become resistant to the GeneDrive, and then be able to attach itself again to the mosquito. Maybe this time the virus is going to be more dangerous than before, and people are going to become more sick than before, and maybe more populations are going to be affected than before. That's one case. The other case, it would be with no mosquitoes around, there would probably be other species that feed on human blood who would have more resources, blood, to feed on, and their populations would increase. One possible case is the biting midget that feeds on human blood. Humans wouldn't have to worry about mosquitoes anymore, but this time they would have to worry about other species, such as the biting midget, who could potentially bring new, more dangerous diseases that people aren't ready for. The other issue is with our natural food chains. When we remove one species from the ecosystem, that can have negative consequences to the food chain. Mosquitoes, for example. Who eats mosquitoes in the food chain? One example is the Ebarcha culcivora spider, better known as the vampire spider, that feeds indirectly on human blood by eating female mosquitoes that carry blood. If we remove mosquitoes from the equation, what would the spider feed on? 80% of its food is mosquitoes. Then what would happen to the species next, to the predators of this spider, and to the predators of these other species? How would the entire food chain be affected by this? We need to be very careful with the applications that we use. One case that could probably happen when there are no more mosquitoes, the spider could probably start feeding on other species that feed on human blood, such as the biting midget. Another case, and this is just hypothetical, could be that the spider could evolve to be able to feed directly from human blood. For this, it would need to evolve to have a skin-piercing mouth to be able to bite skin of humans, the same way that mosquitoes have. This is just a hypothesis. In the case of mice, if there are no more female mice, and there are only male mice, we don't know if they would interbreed with other species, and if that action would affect negatively native species in the area. Maybe it interbreeds with a native species that wasn't targeted by scientists, and that we want to live, but because of this interbreeding, the species is going to go extinct because it's going to produce only male offspring. So we need to be careful with the application that we have on this technology. Okay, at the beginning I mentioned that gene drive research currently works with the sexual reproductive species that reproduce rapidly. What if, in a few years, when this technology has become more sophisticated and scientists are able to understand all the little bits and pieces of the technology, what could happen if we start experimenting with humans to enhance our species, make superhumans? Do you want to be taller? We'll do it for you. Do you want to be more intelligent? We'll do it for you. Do you want to be free from any diseases? Sure, we'll provide it to you. This makes me think of Berejnoi. In this book, humans live in a pain-free society, and humans are engineered in artificial wombs. Everything seems to be perfect, but sometimes a perfect world isn't so perfect. We don't know how that would affect our cultures, our diversity. We have blind people, deaf people, people with autism, or any other people who have different abilities from the norm. We could affect this diversity that we have right now. And that's what scares me. Because when we start dealing with our natural genetic composition, there is no way of coming back. So we need to be very careful with this. So now, let me go back to the beginning. Gene drive research. Is it ethical? Is it unethical? I don't want to say it's plainly good or bad, because it can be both and neither. The goodness and the badness is going to come from the specific use and purpose of who handles this technology. It could be used as a weapon, or also as a tool to eradicate malaria, Zika, dengue, chikungunya. So it depends on the source, on the end goal. There are people who support and who oppose this technology. The Bill and Melinda Gates Foundation, the UN are examples who support the technology. One organization that opposes the technology is the International Union for the Conservation of Nature. Some individuals who oppose it include Jen Goodall, Vandana Shiva, Dr. David Suzuki, who is a genetics professor and broadcaster, and many others. This technology has pros and cons, and we need to be aware of that. And even though I fear the negative consequences of gene drives, I can't hide the fact that I'm also curious of what the consequences will be. Will it benefit humanity in the long term? Will it change the course of our natural food chain? Will it lead to the next species of humans? I don't know. I just want to hope for the best. What I do know is that more research is necessary. We need to look at the consequences, at the political, regulatory, ethical, social consequences. We also need to include all the stakeholders who are going to be affected by this technology and who are going to have an effect on this technology. We need to be inclusive. So with that, I'm going to end. But I want to leave you with this question. If the future of gene drive research were in your hands, what would you do? Would you support it or oppose it? Thank you.